You know, that very first connection we ever make, that bond between an infant and its mother? We tend to think of it as this magical, totally unbreakable force, right? But what if I told you it's actually more of a delicate learning process, one that can be seriously disrupted? Well, today we're going to dive into the powerful and, yes, surprisingly fragile science of attachment. All right, so let's start right at the beginning. This bond is the foundation for pretty much everything that comes later. So the big question is, how is it actually built? And that really brings us to the heart of what we're talking about today. Is this bond just automatic? A biological instinct that just flicks on the moment a baby is born? Or is it something that has to be learned? Something that needs to be actively reinforced in those first crucial days of life? Well, for a lot of species, including the rats in the study we're looking at, it is absolutely a learning process. They call it attachment learning. See, a newborn pup doesn't just magically know its mother. It has to memorize her specific, unique scent. That scent becomes everything. It's a lifeline, really. It guides the pup to the only source of food, warmth, and safety it has. So this isn't just about some fuzzy feeling of love. We're talking about pure survival here. Okay, so if this bond is something that has to be learned, it begs a really important question. What happens if you mess with that learning process? What if the world is totally unstable right at the exact moment that critical learning is supposed to happen? Well, that's precisely what a group of scientists decided to find out. And just to be clear, this wasn't just some abstract experiment about rat pups. No, the researchers had a much bigger goal. They wanted to create a model that could help us understand really complex and really important human situations. I'm talking about things like early adoption, or what happens when an infant has to deal with frequent changes in their caregivers. This whole experiment was designed to see what that kind of early instability does to the most fundamental bond we have. So here's how they set up the experiment. First, you've got your control group. Super simple. The pups just stay with their biological mom. That's our baseline for what a stable bond looks like. But then things get really interesting. You have the early RCF group. These little guys had their world turned upside down right from day one. They were swapped between different mothers for their first 3 days of life. The huge question for them was, could they even form a bond at all? And finally, there was the late RCF group. They got a normal, stable start, but then they were swapped between moms on days 9 through 11. This was designed to answer a totally different question. Once that bond is already formed, is it strong enough to handle that kind of disruption? Now, this whole procedure is called repeated cross-fostering, or RCF for short. And yeah, it is every bit as disruptive as it sounds. Just try to imagine this. Every single day, for 3 days in a row, a mother is taken away and her entire litter of pups is switched with another one. For these newborn pups, this created a world of total chaos and constant change, right at the time when they needed consistency more than anything. So, after all this disruption, it was time for the moment of truth. The big test. You can't exactly ask a rat pup if it recognizes its mother, right? So how do you find out? You test its preference for her scent. This was it. The big reveal. Did these pups, after everything they went through, actually learn who their caregiver was? And you know what? The first result was something of a surprise. It wasn't even about the pups at all. It was about the mothers. The data showed that the moms who were part of the swapping process showed way less maternal care. Significantly less. So the whole thing was clearly super stressful for them too, which of course just made the environment for the pups even more unstable. Okay, but here it is, the first really crucial finding. Let's look at those pups from the early disruption group. When they were tested on day 7, they showed absolutely zero preference for their caregiver's scent. Look at this. They spent just as much time near the mother's scent as they did near plain, clean bedding. It was a total wash. The constant swapping, that early chaos, had completely stopped the attachment bond from ever forming. Plain and simple, they didn't know who their mother was. But now, check out the results for the late disruption group. This is where it all clicks into place. It's a completely different story. These pups, the ones who were swapped later, showed a strong, unmistakable preference for their caregiver's scent. Think about that. They went through the exact same disruptive swapping procedure, but because their bond had already formed before it all started, it held strong. The attachment, once it was locked in, was resilient. So what gives? Why the completely opposite results? One group fails to form a bond at all, and the other's bond is rock solid.
Well, the answer comes down to one single critical factor: timing. And it all becomes clear when you look at this timeline. See, scientists have identified something called an attachment-sensitive period. For these rat pups, it's basically the first 10 days of their life. Now look at the experiment again. The early disruption, it happens completely inside that critical window. But the late disruption, that happens right as the window is closing. So here's the main takeaway. This sensitive period is like a biological window of opportunity. It's this special time when an infant's brain is just perfectly primed, totally ready to learn and lock onto the scent of its caregiver. If you disrupt that learning process during that open window, the bond might just not form. But if that bond is already formed and the window closes before the disruption hits, it's strong enough to weather the storm. Okay, so this is about way more than just a fascinating look into the world of rat pups, right? These findings have some really profound implications for how we should think about early development in all mammals, and that includes us. So let's just boil this all down. What are the big takeaways here? Number one, a stable, consistent environment during that sensitive period is not just nice to have. It's absolutely essential. Two, instability during that window isn't just a minor setback. It can literally stop that primary bond from ever forming. Three, once that attachment is formed, it becomes way more resilient to later disruptions. And finally, and this one is huge, this isn't just about being a baby. This very first bond acts as the blueprint, the model for all of our future social behaviors. You know, the researchers themselves really said it best in their paper. Here's a quote. The same postnatal environmental intervention at different times in development may induce different or even opposing effects. It sounds a little academic, but what they're saying is powerful. It means that doing the exact same thing at two different points in early life can have completely opposite results. In other words, when it comes to building the foundations of who we are, timing isn't just a big deal. It's everything. So this research on rat pups really forces us to look in the mirror and ask some much bigger questions about ourselves, doesn't it? It just hammers home the incredible importance of stability and consistency in those first few days, weeks, and months of life. And it really leaves us with a vital question to think about. How can we, as a society, as communities, as individuals, do a better job of protecting and ensuring that stability during life's most sensitive and most formative periods?